I went through the whole thing. But he is at the University of Uppsala in Sweden. He has his PhD neuroscience from Banaras University. So Banaras, one of the oldest cities in India. It's now Banaras city. I beg your pardon?
In the world.
Exactly. Exactly.
Living agency.
So, and he is, I'll leave it at that. Please have a look at Dr. Sharma's blog.
What is this like? Okay. Okay, thank you very much for this honor to be here and speaking to this audience comprising disciplines of physics and chemistry and biology and working on nanomedicine. So naturally, maybe some confusion may arise, and I'm here to hear your views. We have a long project on nanomedicine supported by European Office of Aerospace Research and Development, Department of Defense, US government, and together with different organizations, the Swedish Strategic Research Foundation. I work in Uppsala University, and this is US Air Force Research Laboratory, and we have collaboration from Basque Government of Science Foundation, and also this Romanian Government of Science Foundation. So I will present you today some of our experimental results on nanodelivery of cerebrolysin, a drug, on experimental Alzheimer disease. But before I should go that, I want to tell you that all views are mine. It has nothing to do with any other government with whom I am working. We are also initiated some very large project with Chinese government, Ministry of Science and Technology, with several, it's a very generous grant on Chinese herbal medicine, but I will show you maybe the next time. So for some of you, I want to show that this German scientist was the first to discover Alzheimer, and this is the lady patient, and after his name, the disease was known as Alzheimer's disease. Actually, what happens, everyone can have, and there is the question that whether this Alzheimer is the cause of disease or it's accelerated aging. We do not know, but I'm a neuropathologist by training, so you can see that this is the normal human brain, and after Alzheimer, it can have like this. So naturally, we have different kinds of problems, and this is showing an example with PET scan that a person having Alzheimer's disease have dysfunction in temporal lobe. You might be knowing about this, and the pathological aspect is that there is a deposition of different kinds of pathological things, amyloid plaques, for example, and they disturb the connections between neurons in the brain that is causing the basic aspect of the disease. Here is one example of deposition of amyloid beta in human cases of pathology in the cortex. And actually, this is a cartoon taken from the literature where we have this microvascular system. These are neurons, and the glial cells all are affected after Alzheimer's disease. Going to intracellular, there are several proteins were affected, and one of the important proteins is tau protein that is severely affected, and this could be considered as one of the causes of having this plaque deposition. There is also a cartoon that why different enzymes were denatured and resulted in beta amyloid plaque deposition in the brain. There are two different theories in Alzheimer cases, that whether this is the deposition can cause the disease, or probably our nervous system, when it is diseased, can't take out these from our brain. But the theory is still controversial. This is also senile plaque human cases, and you can see that severe damage in the brain. But we are working on the aspect of brain and circulation, because that's the cause of dementia. Either vascular dementia can lead to the disease, and this is based on the fact that we have, of course, about 10 billion neurons in the human brain, but we have even much more glial cells in the brain, and obviously endothelial cells were even higher. So when we talk about neuroprotection, I think we should focus on all these three elements, not only neurons. I had an interest in blood-brain barrier since a long time. In 1992, this was my first PhD thesis in Banaras Hindu University. And after that, we focused on this research supported by various organizations and different governments in EU and United States. And we believe that blood-brain barrier could be the gateway of diseases. Here is a cartoon showing that when this capillary that consists blood-brain barrier is leaky, it can influence these glial cells and neurons, causing disease. One of the finest examples was given by Dr. Brightman and Rees at National Institute of Health in early '69. Here is the lanthanum. It's 12 angstrom in diameter, and it was seen stopped between the two endothelial cells. Here is the tight junction. So this was the first anatomical evidence of the blood-brain barrier. But now we know that also these endothelial cell membrane are infiltrated in disease processes, and obviously also in Alzheimer's disease. So we replicated this model in our laboratory in Sweden, and here is lanthanum, 12 angstrom in diameter. This is a rat spinal cord, and the tracer is present inside the lumen, and therefore the neurons and glial cells were completely intact. I am a morphologist, so I am showing some examples of the light and electron microscopy. So we work on the theory that any insult to the CNS opens the blood-brain barrier. It has also a spinal cord, so therefore it is a blood-CNS barrier. It can, when this is disrupted, one can have gene expression, apoptosis, edema, and all these together can lead to cell death. Now our point is that whether we try to interfere at this level or this level to have this cell death reduced, resulting in neuroprotection, we are still working on that. Concerning Alzheimer's disease, I can show you some data, and you can see that this is in 2007. We know that glutamate is one of the neurotransmitters that is excitotoxic, and here you can see that these glutaminergic transmission is altered. There are some examples that even post-mortem study of amino acid neurotransmitters, they are altered, and we have this excitatory amino acid glutamate, and we have inhibitory like GABA and glycine, and aspartate is excitatory. So there is a balance. We need balance in this situation. But this balance is increase of excitatory amino acids and decrease in inhibitory amino acids are disturbing. We are using a drug called Cerebrolysin because when in the brain we have several different neurochemicals and factors. So it is almost impossible to think that one particular drug or compound can do all neuroprotection. Cerebrolysin is a balanced composition of several neurotrophic factors and active peptide fragments. And there are, this is clinically approved by FDA, and there are several trials going on in the United States as well. So here the cartoon shows that some of the important part is amyloid beta peptide and tau protein. They are working in dementia and also neurodegeneration. So we use this drug called Cerebrolysin. It is manufactured in Austria, and it has a multimodal drug used in brain injury and Parkinson's disease and neurodegenerative changes. We are using experimental models of Alzheimer's disease. Some experimental evidence shows that this is neuroprotection. Traveling in US Air Force planes and names and even our officers are breathing almost every day, every parts of the world. So the question is whether the same kind of injury is occurring in those people. I think the pathophysiological response and even the drug response will be different. So the question is, why nanomedicine? The point is that we have evidences that if we deliver the drug, any drug, I mean that is good, with nano delivery it can have better effect. It may have long-lasting effects. It could rapidly penetrate into the central nervous system, and it is less degradable, and its distribution is quite widespread. What about the neurotoxicity? And that is one of the important problems. Still, we are working on that. The question is, here in this audience, you know very well that the drugs can be delivered by various ways, and we have just heard a nice presentation by the previous speaker. So the point is that whether if we deliver the drug using different type of nanomaterials can have different effect. The second point is that what is happening neurotoxicity versus neuroprotection. We still do not know the neuropathological aspect of nanomedicine, and that we are working together with US FDA and other government organizations. To make aware, we have published this progress in brain research earlier in 2009, that we have established the discipline called nanoneuroscience and nanoneuropharmacology, that we need good attention. This is my good friend Dr. Ryan Tian. He has the US patent on titanium nanowires in terms of scaffolds. So we are using his technique, and this is Dr. Wang. Both are having the US patent and this titanium nanowire, and we are using drug delivery bound to these titanium nanowires. Of course, their titanium is, to some it is neurotoxic, but in our hand, when we use titanium nanowires in normal animals, and there are certain literatures published in Springer journals, I do not have the reference at the moment, it is least damaging and totally innocuous in our hand. The recent trend also suggests that we are talking about so much stem cells, and there are reports that stem cells can also be delivered using nanowires, and when it is done, they live longer. This is an example, and they can be in the system for a long time, and their effect is much more better than the stem cell delivered alone. So this is the nanowire, titanium nanowire we are using from Dr. Ryan Tian's lab, because the technology is done there, we are only using. And you might be knowing that every year there is National Innovation Summit in the United States, organized by the government of the United States and several all defense research organizations. In this year, in 2016, it will be in May in National Harbor, and the last date for submitting abstract is December 11. So if you have any technical innovation or any kind of innovation, you should go there. When we submitted one of our innovation with Cerebrolysin, we are surprised, but we are very pleased that our nano delivery of Cerebrolysin in neuropathic pain was considered top 20% of technological innovation in 2013. And this research also came in various spotlights in US government journals. So this is our basic model, and Dr. Tian and laboratory, they use it. So what we did for experimental model of spinal Alzheimer disease, you know that this is the deposition of amyloid beta peptide. So one of the model is that you infuse amyloid beta peptide in lower doses in the brain for 4 weeks. This is the standard model, and then you see that neuropathology. So here is the case. When we infuse in the brain, in the normal, it's not so much. And then here we have used, these are the results on knockout mice, where you can see that A-beta deposition is very high. When we have given Cerebrolysin for 4 weeks, you can see that this level is reduced. When we have given nanowire Cerebrolysin, you can see we have much better results. Of course, until 6 weeks after that. We are not sure that if we stop this drug, we can still continue after more than 3 months. So another example is tau protein. Here you can see, this is the normal cases. It is increased in knockout mice, and Cerebrolysin alone is doing better, but nanowire Cerebrolysin is doing much better. Here are some histopathological studies, and you can see that this is we are looking across the vessel, and this after A beta infusion, we can see amyloid deposit, that is obvious, but nanowired cerebrolysin has really reduced this deposition even after infusion. This is cerebrolysin normal condition. And here you can see, when we believe that blood-brain barrier could be the basic aspect of all disease, we have measured albumin, because in our blood we have albumin, and when there is a leakage, it goes into the brain and we can detect by immunohistochemistry. So here you can see that albumin deposition were high, and nanowired cerebrolysin is much less, and normal cerebrolysin is also less, but this situation is much better. About GFAP, here you can see that you can see here that in normal situation it is much more highly activated, but nanowired cerebrolysin has the reduced effect of activation of GFAP, means inflammation, and cerebrolysin is also good. Coming to neuronal study, this is Nissl staining, but you can see that nanowired cerebrolysin is the best as compared to the untreated group and even cerebrolysin. I did some basic data here, and now I can tell you that what is happening. All experiments done in physiological or neurological laboratory on normal human beings or normal animals, for example. But nowadays, this Alzheimer or any other condition, we are all human beings, we have either hypertension or diabetes, but no experiment or experimental drugs were tested in animals having comorbidity factor. So this is our long-term plan to study what is happening of inherent disease. I'm showing this in diabetic animals. When we use this diabetic animals of the same amount of A beta peptide, you can see here that the blood-brain barrier leakage to Evans blue was very high, also to the iodine, neural injury, number of cells, and astrocytic activation. These are rough estimation. And they are also showing some effect of the dose, 20 microgram versus 10 microgram. And this is the diabetes alone. Then, when we have given cerebrolysin, in this situation, what we need to have higher dose of cerebrolysin if it is normal cerebrolysin, in cases of diabetes. So the point is that when we have multiple factors, then we need to revise our drug doses, or we give nanowired cerebrolysin. Even in 5 milliliter per kilogram dose, we have the highest level of neuroprotection. And to confirm at the histological level, you can also see that the aggravation of pathology is much higher, both neuronal and astrocytic and albumin deposition, sorry about that. But this nanowire delivery is much better in these situations. How to get away with this? Anyway. Now I am telling you in last few minutes the effect of hypertension. We have given this peptide in normal condition and also the hypertensive, and you can see that leakage was much higher, and brain edema was also higher in hypertensive group after amyloid beta peptide infusion. The point is that if we are going to deliver in 2 different modes of the same drug, we have one, I showed you Nanowired, the another we have done in Italy, and this PLGA level cerebrolysin, you can see that effect is still better here, but the nanowired cerebrolysin appears to be a bit superior than PLGA delivered cerebrolysin in same doses. The same thing is also happening in neuronal injury. Hypertensive animals showed higher injury, and nanowired cerebrolysin was the best. Also this albumin deposition. This is a crowded slide, but I just want to show you that this is the titanium nanowired cerebrolysin delivery, and this is PLGA delivered cerebrolysin. But you can see here that although PLGA delivered cerebrolysin was also quite effective, but it appears that titanium nanowired delivery of cerebrolysin is much more better. There are different regions of the brain. And these are the examples, final examples, that You can see that cerebrolysin given, here is the deposition of A beta peptide, and here you can see that the nanowired cerebrolysin is much more better. This is the albumin deposition, is also much better. And these are some data showing that in different brain areas, the nanowired cerebrolysin is much more doing good balance between excitatory and inhibitory amino acid than PLGA. However, they all are quite effective. So now we are working with Dr. Andrew Schell. He's a Nobel Prize winner in physiology and medicine in 1977, and he has CRF fragments. We are trying to nano deliver these fragments to see alone or together with cerebrolysin, can we have much more better potentiation of these effects. And in this case, I must acknowledge the previous governor of Arkansas, who is helping us a lot. And even he is not the governor, but we get lots of support with him. Finally, now we are working on this titinate nanosphere, and this is done by our colleague in Arkansas, Dr. Asia, and we have she has injected cerebrolysin in these nanospheres, and you can see here that the drug release is quite good after 3 hours, and we have also the higher data on this. So the cerebrolysin is delivered like a constant infusion pump, and this is the, and she presented this paper in Parkinson's disease in Society for Neuroscience in Chicago a few weeks before, on 21st of October, and this was done a special mention in Society for Neuroscience, where we have more than 31,000 abstracts. Also, the PLGA loaded cerebrolysin, our friends in Italy have analyzed its deposition and storage, and you can see that the cerebrolysin was released. This is 1400 minute, and even the drug level with PLGA was kept at 4 degree or 24 degree, even 150 weeks. They were, 150 days, they are quite stable. So the drug was quite stable, and some of you might be knowing Dr. Croto. We have some interaction with him, and probably we'll work something together in his laboratory. Now he's in London, UK. And so the point is that I gave 2 messages. First, that same drug can be quite effective if delivered with nanotechnology, but however the different nanotechnologies applied, the same drug can have a little different effect. And finally, no bad drug can be termed good if given by nanotechnology. The drug should be good. We can't change its properties, but maybe enhance its neuroprotective abilities. And of course, this is multinational cooperation. I especially mention here the International Peace Hospital in Shijiazhuang in China, and US FDA. There are several organizations supporting us. I'm not going through them. We have lots of collaborators, and thank you very much for your kind attention.
Question? Dr. Sharma? Right there.
Yes, please.
Yeah, Professor, the human system has amazing. The human system has amazing defense mechanisms that recognizes any kind of foreign materials that enters into the body for elimination. So I think this is the major challenges in nanomedicine to deliver drugs to a targeted cells. So is there any measures that we take to boycott this defense system in order to reach the targeted cells?
Yes, your concerns are quite genuine, and in Stockholm University, they even deliver nano-level antibodies to detect some type of cancers intravenously. In Arkansas, they use this titanium nanowire level drugs and deposit after knee operation, and the regeneration is faster. So obviously, every drug has some problem. Otherwise, we have anti-cholesterol drug. People are not dying with that. They are given for good purpose, but there are so many side effects. We live with that. So likewise, nanomedicine is also have the same challenge. And its clearance is also very difficult. It takes long time, you know. But that's why we are choosing the least innocuous nanoparticles to deliver drug in order to get some better effect. But obviously these challenges are very similar to any kind of pharmacological drug we are having at the moment, with or without nanomedicine.
Thank you.
Excellent talk, Dr. Sharma. Thank you very much. So I think you'd like to take our picture as I present this to you.
Sure. Thank you, Mr. Sharma.